Hey guys, so I decided to post another recording uh, just for chapter 21 because I think there is some stuff in here that's worth going over, at least fairly quickly. Um, so the general idea of chapter 21, or at least the stuff that you guys haven't heard before, is how how genetic change is actually going to take place over the generations. Um, Chapter 22 discusses a little bit um, how genetic change occurs normally, but there are mutations that can occur which are going to um, sort of exacerbate the degree of change happening within the genome. So during sexual reproduction, you have processes that um, provide the offspring with a new genetic code that is different from its parents. The very act of sexual reproduction is creating um, an individual with genes from the mother and genes from the father, but there's also events like uh, crossing over, which introduce a bit of randomness to the offspring so that you don't have um, two brothers that are genetically identical. Uh, but in meiosis, there's also a lot of opportunities for mutations to occur. And the the most common way for this to happen is through duplications and inversions, um, so that you can have, like with the human chromosome relative to the chimpanzee chromosome, we can see that the human chromosome is a duplicated section here. It's got this telomere-like sequence in the middle because something happened um, during among our common ancestors that we have with chimpanzees, and something happened to have a duplication, so we have uh, a longer chromosome. Uh, Inversions can also occur. The rate of duplications and inversions accelerated about 100 million years ago when dinosaurs went extinct and mammals diversified to fill all the new niches that were available. And they were breeding a lot because there were more resources, um, more places for them to move into, and that led to um, a higher rate of duplications and inversions because there's more offspring being reproduced. And so producing more offspring led to species diversification faster because of just sort of the rate of random mutation. You can also have unequal crossing over, which can result in uh, duplications and deletions. So see, they crossed over at the wrong spot here, and so now we have a chromatid with a duplicated gene and a chromatid with a deleted gene. We can also have rearrangements of gene parts, like an exon duplication and shuffling. The movement of transposons around too is going to contribute to genome evolution. And these can these also occur in meiosis. So they're going to be affected by the rate of reproduction as well. Uh, when you look at the genome of an organism, organisms with a larger genome are going to have more non-coding material there, um, more exons, more transposons, and therefore more potential for genetic diversification. Um, there are more cards to shuffle around versus an organism that has a smaller genome, has less cards, and is going to have less opportunity for them to be put in a different order. So over time, we've seen some species get a lot of genetic material, but genetic genome size doesn't necessarily correlate to our ideas of evolutionary progress. And there's really, when we're talking about evolution, there's no, we are no more evolutionarily successful than bacteria. The bacteria are suited to their environment, we're suited to ours. So we're both pretty successful. So this is talking about the transposons that I mentioned earlier. I'd like to move down and talk about the, where are we at here? Oh, the homeobox. This is a good thing for us to touch on. Essentially, the homeobox, or the Hox gene, is a highly conserved gene among eukaryotes, but it's also present in some prokaryotes. And it essentially is responsible for the regions of development from an embryo to an adult organism. So you can see that, you know, this green section in the embryo of a fruit fly correlates to this green section on the head. Brown goes to legs, orange goes to abdomen. And this genetic expression works the same way in a mouse. So the homeobox, or Hox gene, is responsible for the ordered development of an organism. And the reason that a gene like this is highly served is because it's essential for proper development. You can't have an individual grow to maturity and reproduce with something seriously wrong in the Hox gene or the deletion of it. So it's highly conserved because any organism that had a mutation in the Hox gene, that organism was an evolutionary dead end because it would have died or been unable to reproduce. Or even if it somehow was able to live and reproduce, it certainly wouldn't have done so well as the ones with a normal functioning Hox gene, and therefore that mutation would have been lost in the gene pool rather quickly. And there's a lot of these highly conserved genes. A lot of them are responsible for development. Here's a couple other organisms and their expression of Hox genes. So the real takeaway here from chapter 21 is that there's multiple ways that you can have pretty drastic genetic change through mutations, and most of those have to do with meiosis and errors in reproduction. But if you have a deleterious mutation in an important gene that's highly conserved, then you're probably not going to have that organism survive to reproduce. There are, of course, more minor mutations that can occur, like y'all should have learned about SNPs before, but all the drastic genetic change, like an extra chromosome being created, that's going to happen due to errors in meiosis, most likely. So I hope you guys, I hope this was helpful for you. I get the feeling this is mostly stuff that you have been over in Bio 111, but I thought it might be helpful to go back over some of this stuff again. If you have any questions, be sure and let me know. We'll be moving on to some more fun stuff next week. Chapter 23 is pretty great. Moving into more ecology-focused stuff, which is also much easier to grasp. So thanks for tuning in. I will see you guys next week.